Well, thank you very much. First of all, I want to thank the organizers for providing me this forum. Very appropriate forum, particularly for me this year, as you'll see. And of course, I'm honored to be asked to speak. What I'm going to speak about today is very connected with the program that's running at the Simons this semester. And I should say that this year I've been on sabbatical in Berkeley, and as part of the Miller Institute and as part of the Simons Institute, and both have been wonderful delight to be involved in, and it's a wonderful opportunity for me this semester. And for those of you who haven't gotten involved yet but might be interested, there's one more workshop coming up on the 28th that promises to be quite interesting, especially if you're interested in stochastic processes and probabilistic models. This will be quite a good group. And besides these workshops, there are lots of interesting seminars and reading groups and so forth, and most importantly, lots of interactions, interpersonal interactions at the actual Simons Institute among students and postdocs and visiting faculty. So this has been very successful from my standpoint. Okay. Well, I had a little bit of a challenge for myself. This semester has allowed me to think about a lot of different things, and the program has gotten me thinking even more broadly. And so when I got around to thinking about what I would talk about today, I found the challenge of a sort of more open lecture exciting and interesting, although, of course, I ended up very quickly not being able to get everything in that I thought should be. Anyway, this is what I intend to cover today. I hope I can make it quickly enough. The first topic is not head-on other than motivational, having to do with just making sure we're all on the same page about what has happened in the last, say, less than a decade in sequencing and what the consequences of that are. Well, at least one example that I'm particularly familiar with. And then I want to launch into my thesis about this general topic of evolutionary genomics, what's sort of unique about the history of the field, and particularly appropriate for those people who are interested in quantitative analysis, in inference, and strong tools in areas where there are big problems. I think population genomics is one of those, and the history of it throws a strong light on that. I want to point out just a few of the interesting problems or questions and contradictions that already exist and are becoming more and more serious for the field and have to be addressed with the new data. And finally, I will spend a little bit of time at the end showing a couple examples of how to one might address these contradictions with some more recent full population genomic sequencing data, and also some interesting questions about the scale of natural selection. In other words, what magnitude of natural selection is actually important in some recent experiments that I've been involved in to detect really surprisingly important but weak forces operating. Okay, so getting right along with it, I'll start off with a slide that I wouldn't be surprised that most of you have seen, probably forgotten, but it's widely touted by people in the genomics field, especially the NIH, NHGRI. This is this plot of the cost of, and for those of you in computer science, you'll recognize the sort of poking that's involved here. This is the costs per base pair or per megabase of sequencing over time. And during the Human Genome Project, this line went down pretty much as Moore's Law, both because many of the problems that were being solved were similar in some crude way with those involved in the computer industry. But more importantly, people discovered very quickly that computers were probably, if not the most important part of genomic sequencing, they were certainly the second most important part of genomic sequencing. And so it was following the course of the resources that they actually had available. And then about 7 years ago, there was a very rapid drop. After the Human Genome Project was finished, a number of new technologies came out, which are euphemistically known as next generation technologies. And the price per base dropped very quickly. And you can see now it's leveled out again, probably following Moore's Law more or less. And the consequence of this technology shift which I'll discuss a little bit in detail in a minute, is broad and very far. But most importantly, it enables population genomics to actually take place in a real setting. And it's happened most profoundly in human population genetics because that's where the resources and the goals were initially for the NIH and the Wellcome Trust and many other funders. Okay, but I want to tell you about the consequence of that change in technology for genomic sequencing. In other words, the human genome was finished, but there are genomes that have not been sequenced, many, and some of them have not been sequenced for very good reasons. And one of those are the members of a very important, not that big a clade, but very old clade of conifers. These are pine trees and junipers and so forth, gymnosperms in general. These are very ancient plants, seed plants, and they have a very important role in terrestrial ecology, but no one has even dreamed of trying to sequence these genomes for very good reasons. The genome size is 8 times human. No one thinks these days that the number of genes in these kind of organisms will be 8 times as big, so that means the increase in the genome size is largely going to be repetitive. It turns out it is. So that you have this enormous amount of repetitive DNA, even though the karyotype, the actual chromosomes of the organism looked at through the microscope, seem to be quite conservative. It's chock full of repetitive sequences. We had an inkling, and it turns out to be a good hunch, that most of these repeats are quite ancient, or ancient enough that you can distinguish them at the scale that one wants to do next generation sequencing. And the cost of next generation sequencing now is so low that the actual cost of sequencing such a huge genome to quite good depth is nothing compared to the cost of actually trying to assemble it and understand it. I mean, it's just it's not even relevant, actually. And much more important is actually how you're going to do it rather than the cost of sequencing. Now, the tools for assembling such a genome are really not, we already know right out of the box they're not going to work for 2 good reasons. One is all of these repeats are going to be a huge problem for using assembly techniques that depend on, say, euphemistically, people talk about De Bruijn graphs or some very leveraged string assembly basis that is straight ahead. Those have big problems with repeats, and they have big problems with errors. And the classic overlap layout consensus approach really doesn't scale computationally for this big of a genome because we're talking about so much data that actually to apply the sort of classic algorithm that, for example, is used with the human genome would not be really possible. So one needs to come up with a better approach. And the approach is to do some front-end reduction of the size of the input data. And to do that, we struggled to try to think of what the most powerful ways to do this are. But before I do that, I just want to remind a few of you who don't remember or have never bothered, and maybe it's a good reason not to bother because it may all go away eventually, what the sequencing approach is that's used today generally, and it's been proven quite strong technique, which is just to take all the DNA in the genome, shear it up into small pieces, and then this is the genome here, we're going to shear it into these small pieces, and then we have a technology that allows us to read the sequence from each end. Depending on the size, you might be able to read all the way through, but most of the time not. You have some sequence on the end of these things, and then you ask a computer using one or another algorithm to try to find a way to assemble these into contigs. Sometimes they're gaps. You hope you can find another technology that will bridge those gaps and make those contigs into scaffolds. So we tried to use this technology, but as I said, we had to do some front-end data reduction, if you will. I use the word reduction in a sort of, I want to be more explicit. The first one is biological. Unlike humans and unlike most animals and actually most plants, angiosperms, gymnosperms, and especially conifers have a very unique biology that we can leverage very strongly in this situation. I don't know when the last time you ate it, but very often, if you're eating, especially in the Bay Area, you will run into this thing right here on your dinner plate. This is a pine nut. Okay, so there's something very unusual about a pine nut in biology. There are very few tissues in multicellular organisms plants or animals, where you can get actually a significant amount of tissue that is haploid. In other words, it only has the genome from mom, not from dad. And so the nut part of a pine nut, which is most of the pine nut, can actually be isolated and the DNA extracted from that small pine nut. And there's enough DNA using sort of technologies that we used 2 or 3 years ago, but now we could even do more, to build a number of libraries from that one genome that are deep enough to actually sequence very, very deeply in that one haploid genome. And the role of sequencing deeply you'll appreciate in just a second, and that is we got 75 billion reads, or molecules were sequenced on both ends. And the first thing that we would do is take we identify all the k-mers, say 20 formers in the whole data set, and we would only allow molecules to go through where that particular sequence, where those k-mers in that sequence were seen at least twice in the data. So anything that was seen once was pitched out. So that's very strong filtering for errors. It works quite well. You get a huge reduction in the data and reduction in error rate. And then after that, we used what I would say, it's already published. I didn't do this, so my colleagues generated this algorithm, which is a fairly heuristic algorithm for generating what they call super reads, which are basically you start off with a paired-in read, and then you try to extend the paired-in read with another paired-in molecule, and you end up with the largest things that have at each end a paired-in read, and the sequence in between is completely filled in. And those are called super reads. The actual algorithm is fairly heuristic. It probably could use some rigorization, but it, again, creates a very big reduction now in the amount of data, going from billions of things to 150 million super reads. Now, these molecules, or these constructs, are about 500 to 600, 700 base pairs long. Some are longer. But they are a size that will then, and then a number that can go into a classic overlay layout consensus algorithm. This is just the son of, or the grandson of Gene Meyer's program that was used for the Human Genome Project. We feed it into there and didn't do the usual sort of messy things that people do with genomic assembly. After you do that layout assembly, you go back and try to fill in gaps sort of post hoc with additional sequence. And then we did some more scaffolding using RNAs and so forth. And that produced, on one hand, a very good genome. It's almost the size estimated from both cytology and from simple k-mer counting. It looks like we have over 93, 92% of the genome is in contigs. And the average size of a contig, though, is still small, 8,000. The scaffolds are about 67,000. And so there's still a lot to do, but the quality seems to be quite good. We compared it with some clones that were sequenced from the genome and assembled, and we got very good coverage of the clones. That means probably not a lot is missing. And we got very good identity of the clones in the sense of very similar sequence identity. But notice that the 2 megabase scaffolds that we have here, 2 million scaffolds that we have in this assembly, are a long way from the 12 scaffolds that are actually the genome, which are the 12 chromosomes. In any case, I show you this to show you that there are these emerging problems where the paradigm for doing sequencing is changed by having this enormous power, so that you generate so much data that you actually have to come up with a sort of serious, thoughtful process of pre-processing it into objects that can actually be assembled by the known algorithms or approaches. Okay. Now, that's just to sort of drive home the idea about this technology, next generation sequencing, changing the landscape of what can be done, even for de novo sequencing of genomes. But I want to shift now to population genomics. And as I told you, what I want to start off with in my thesis is this, that population genetics, or the evolutionary genomics, for the first 70 or 80 years of its existence, had very beautiful model and theoretical basis and no data. Now, I can get away with saying no data, even with some of my best friends here in the room who generated a lot of data before we were where we are today, because I tried to generate a lot of that data too, and looking back on it, it was a lot of work, but actually, in the context of modern genomics, it was essentially no data. It was anecdotal, sufficiently anecdotal to keep us excited, but actually, in terms of the big problem, it will appear minor as time goes on. Anyway, let me start off, for those of you who haven't been exposed to this, to give you the sort of quick derivation of the concepts that are important in evolutionary genomics and where they come from. And the basic question and problem really was the one that Darwin posed and tried to address, which is how does diversity arise and spread and be maintained in among biological individuals and populations and species. And typical of his day and age, he was primed to move to a model that basically leveraged the difference in time scales and spatial scales, and he seized on this idea that really it's the process, the interaction that happens among individuals in a population that gives rise to the diversity among taxa. And this picture I just put in there, this is from his actual notebook. This was one of the earliest actual phylogenies that was drawn. You might notice it has some trichotomies and stuff, so his ideas about how speciation took place are sort of interesting. Probably wouldn't get you through your qualifying exam today. Anyway, but the main problem that Darwin had, and he didn't make a big deal out of it, but it's assumed by many scholars to be very obviously painful problem for him, and it was for the whole field, was that the mechanism by which the biological differences were inherited was not understood by Darwin at all. He created a whole chapter in one of his books where he elaborated a theory that no one reads because they don't want to laugh at Darwin, but it was quite clear to him and other people for a number of decades that this was a serious problem, that if you were going to address the validity of his ideas about evolution, you would need to have a better understanding of inheritance. And completely on a different world and a different scale, there was this fellow Mendel who was an avid scientific mind, but a man of not great means, and so using his friends in the church to get an education, which turned out to be largely in the physics department at Vienna, he got a pretty good education, but then he went back to his high school teaching and so forth, and he pursued his interest in plant inheritance, basically, in his own garden in that facility with no support. And after a number of years, he published several papers, one of his most important one, which was, as I understand it, only cited a couple times in the first decade or so. So impact factor is very low. Okay, really. H value or whatever it is really not even worth mentioning. So he was completely ignored, but it's critical to understand what was on there. His mind was open to the idea that maybe inheritance would be essentially discrete or particulate, and that the material might be transmitted in units, discrete units. And so he really was able to embrace this, and probably his trainings in mathematics and combinatorics might have played a role. In any case, after Mendel was rediscovered, people got to work seriously on genetics, and especially the Morgan lab at Columbia started working on chromosome basis of inheritance, and they outlined linkage and its relationship to being the linear array of genes on the chromosome. This is just a little figure here for those of you who don't remember. Genes can be, if genes are on the same chromosome, they can be transmitted in a parental way without recombination. In other words, big A and big B are transmitted just like it was in the parent to the offspring. Or if there's a crossover or recombination, then it can produce these recombinant chromosomes, which this group lady showed was a physical exchange of material. And so this is an important element of the theory that underlies modern evolutionary genomics, is this process of recombination. The other process that's important, of course, that Mendel discovered, luckily for him, most of his genes were unlinked, and so he discovered an independent assortment of genes that are on different chromosomes, so that this locus would segregate independent of A and B. Okay, so the other thing that happened that was important was the understanding of where variation came from. And it was a student of Morgan's Muller who was the first person to actually create mutations sort of by force using X-rays. And he characterized their process. Largely, they had a very random process even when induced. And the ones he worked with were mainly very severe lethal mutations, but largely behaved as points as far as genetic experiments are concerned. They were very discrete. And the idea that there was a mutation rate and it could be, in his case, manipulated became part of the theory. So these ideas formed, and they're very elementary, simple ideas. They laid the groundwork for evolutionary genomics, and they're very simple ideas. And the only thing that needs to be added to that is what happened, how organisms mate. And most population genomics deals with organisms that mate at random because that gives very strong mixing and independent assortment. It makes the equations much simpler to interpret. And I'll just brush quickly through the sort of founding contributions or contributors to the field, but actually there have been hundreds of brilliant people who have contributed lots of wonderful things to this field. Meanwhile, keep in mind, there's no data, essentially. There's a little bit of data on correlation among relatives, but the actual genetic material, nobody knows anything. And so Fisher was the founder and saw how to reconcile quantitative genetic character correlation among relatives of those with Mendelian genetics, and he enunciated a very important theoretical concept that the rate of adaptation is proportional to the variance, the genetic variance in that character. Much work has been done, and in the last few months at the Institute, we've had many discussions of this founding idea and elaborations that have come out of that quantitative genetics. There were many people worked on simple single locus dynamics. One of the earlier contributors, of course, was J.B.S. Haldane, and he's responsible for identifying the simplest relationships between mutation and selection removing deleterious mutations, and mutation putting them in. The person who really went after and was interested and enthusiastic about randomness or stochastic processes was Sewell Wright to begin with, but many other people have followed. And he characterized many of the most important steps that stochastic variation or stochastic change in gene frequencies arise, mainly by binomial sampling associated with what's called genetic drift. But he also enunciated a lot of the relationship between that and the equilibrium distribution of a variation when those processes come to equilibrium. As I said, these guys were sort of started out, and a few other people started laying the groundwork with these very simple ideas. And then there has been like 60 or 70 years of elaboration and investigation of these things. And while some people have said that it's been driven by the new observations of genetic variation, looking back on it now, I think it actually thrived in the absence of data. It meant the theory could be elaborated in any direction with any parameter space that you might want to entertain, and because there was no data really to narrow the scope of what we wanted to do or consider. Okay, so that's the sort of background. Now I want to just leap right up to the present day in population genomics. And as I said, most of the effort over the last 5 or 6 years has been in the human arena, and they've had some spectacularly interesting and wonderful scientific advances based on enormous amount of dollars and IQ point hours and everything else poured into it. And I want to just point out 3 that I think are really quite exciting and exemplary of what we can probably expect to see more of in the coming decade. I should mention the human people have 2 main projects. One was called the HapMap project. That was just basically they went out and collected a bunch of polymorphisms or DNA sequence variants, and they studied just those, not the whole genome. And then following that, the 1,000 Genomes Project was started, where they actually tried to sequence the complete genomes of 1,000 individuals and use that data to make inferences. Most of what I'm going to show you now is based on the HapMap data, although I'm sure more and wonderful things will come from the 1,000 genomes. One of the things that popped out of the earliest data, human population genomics data, was something that looked like this. There were anecdotal stories in the human genetics literature and other literature about there might be recombination hotspots in the human genome, but they were always sort of, oh, well, it's an anecdote, maybe it's true, but it doesn't have any broad genomic consequence. But as soon as people started to look at lots of polymorphisms and they started measuring the correlation between a SNP here and a SNP somewhere else nearby on the genome, and the redness reflects the statistical significance of the correlation in the frequency. In other words, individuals who have, say, the A versus the T at this site are much more likely to have the G versus the C at this site within this block, but not with the site over here, because there's very little correlation between the SNPs in this part of the genome and that. So the scale here is megabases. Megabase or so. So what was obvious right away is that there are these blocks in the hypothesis and interpretation of many people who had worried about this for a long time was almost surely these are hotspots. And immediately people went to the toolbox, got out the population genetics technology that had been developed essentially without data, and applied it very quickly and swiftly using composite likelihood and other methods to estimate. The blue line here are estimates of the recombination rate along the genome. This is unfortunately not exactly the same segment of the genome, but the blue is the estimates from this kind of a data, in other words, from population genetic variation, where you infer from the estimate of the history of the population that there must be a hotspot for recombination at this point in the genome, which would correspond to one of these junctions between the blocks. The red line here is just some very important validation data based on actual genotyping of sperm, showing that, in fact, even in sperm, you can detect these hotspots. There is excess recombination at almost many of the same places, if not all. Okay. What was really, to me, quite spectacular was this happened about 6 or 7 years ago, and within a matter of 3 or 4 years, the groups working on this got to the gene that's actually determining these hotspots. It's a spectacular sort of showing the force of genomic biology to go from basically some pattern in the genetics or some pattern in the population genetics to identifying this gene which binds to a specific sequence which happens to be more abundant in these regions, and that leads to a modification of the chromatin, which then ultimately leads to a crossover at that position. And while the details of how that part of the process goes on, it's pretty clear from work in mouse and humans, both pedigree and genomic data, that this is an important player in determining these hotspots. Okay. Now, the other thing that I think I would say is that many people think, not just I, that really the most obvious and biggest impact of population genetics theory on recent population genomics was that all the theory about how genetic variation in the absence of selection is distributed in time and space, which had been modeled ad nauseum, and when you were a graduate student and you had to read all these papers, there was almost no data, but there was lots of beautiful equations and wonderful ideas, and we couldn't really apply them to much. And so when the human population genomic data came, and now we're getting it from other organisms, there was all this machinery that was ready to roll. And what was remarkable, what this figure shows, is that even the simplest single model you can even think of to ask a question about demographic history is enormously powerful in this setting. So this is a beginning figure from a paper by Lee and Durbin from 2 years ago. And basically they're making an HMM to call segments with different times to the most recent common ancestor based on the frequencies of heterozygous sites here in the genome of one person. I think it's Craig Venter. So if you go along the genome there, you'll find some spots where there are a lot of heterozygous sites, and your HMM will then jump to say, well, there's a much more ancient common ancestor for this segment of the genome than that. So presumably there had to be at least one crossover, because this part of the genome has a different history than this part. And so applying this technology, which is basically sort of off-the-shelf stuff, you can get a very nice prediction of the history, which is the heat map here, and the red is actually simulated data where it's known what the history is, the time of the time to the most recent common ancestor, and you can see that it's quite sensitive, even on a single genome. So this is something you can do with a single genome. Just take Craig Venter's genome or your genome, go across the genome, and you can identify places where it looks like the time to the common ancestor is much deeper or much more recent. And you can average across the genome and learn stuff about human demography as a statistical population thing very quickly. So if you average these things, you can see that the time to here's the recent expansion of humans, the effective population size. The time scale can be converted under the assumption of no selection into time scale of population size, recent very exponential strong expansion. But before that, there's a bottleneck, which partially corresponds to the out of Africa, but even the African populations seem to have gone through a bottleneck. There's some controversy about all that. But in any case, so you can see the expansion in Africa, and then the inferred historical reduction in population size in the African population, and then maybe it increased. There's a lot more going on when one considers multiple genomes. The real problem with multiple genomes is that the computations are not anywhere as straightforward, and much of the discussion, or not much, but many interesting discussions this last semester have revolved around how to extend this analysis that you do just on 2 genomes in one individual to a large sample to use up all the data that we have to make proper inferences about the demography of populations, historical demography of human populations. And I dare say, in the area, we know relatively a lot about the historical demography of human populations, but very often in other settings, ecological and management settings, we know almost nothing about the history of population size and migration in other organisms. And this technology doesn't distinguish between any of those things. It can be directly applied to any organism, any population of organisms. Okay, one last thing. It's cute. You may have heard about it.
Chuck,
yeah.
This video.
What?
The previous figure to make sure the present is on the left.
Oh, no. This is.
No, the next one.
Oh, the next one. Yes. This is the present years ago, 500,000 years ago. 100,000 years ago, 1 million years ago. Excuse me. Years. And this is population size, inferred population size.
Tens of thousands.
Yeah. Okay. Thank you.
What's going on the far right where you saw the curve?
That's probably the thing is breaking down. Most people don't want to talk about that. So but that's an area where that's the part we really want to know about. And so if people can think of much better ways to get at that, the theory is actually they're using very little of the theoretical construct to do this analysis.
So the analysis predicts that there was
no, I'm not sure. I think people don't really believe that.
Yeah. So it's probably about.
Yeah. Here's something cute. So from the beginning of population genetics, there's been a whole way of looking at things where you try to worry about whether the 2 genes that you got from mom and dad, or one gene in your brother and one in you, are identical by descent. In a pedigree system, that's a very well-posed problem. You got genes that are identical by descent, either in the same individual or different individuals. So now in the genomic setting, you can ask, what's the distribution of segments of the genome that are identical by descent because of recent common ancestry? Because people move around and reproduce. And so if you think of this is just a cartoon from the Ralph and Coop paper, and this is one individual who's physically at this position in space. Here's another one over here. This is the distribution of its ancestors over the last 16 generations, over the last 12 generations. And this is the green are the ones for this individual, and the black or gray ones here are the ones that are shared between them at all. The size is the proportion of the genome that's actually would be ancestral, or the probability of it. Actually, it's a probability that any one part of the genome is ancestral in that individual. So anyway, it gives you some idea of the time scale. So we're talking sort of 5 to 20 generations in sort of realistic human parameterizations. And this shows you a pedigree where you can identify the transmission of this little segment of this genome, and this ancestor is transmitted down to here and down to this individual. So those are IBD. And then you'll notice here, this segment is also IBD from this ancestor. So these individuals actually have 2 segments that are IBD. Anyway, these guys then went about characterizing these patterns using genomic polymorphism data from populations throughout Europe. And what you can see here, the sizes of these things reflect the probability of 1 megabase or 1 centimorgan. That's a measure of recombination, part of the genome being shared by 2 individuals in that population. And then the rest of them are the probability of the same thing, a segment of the genome being shared between an individual in this population and any of these other populations. And the color coding is just some identified population location groups, Scandinavians, Eastern European, Western Europeans, and so forth. The thing you can see is that by distance the probability goes down, and that the very close individuals are there. The other part of this is that, which is sort of gets to entertainment genomics, is you can actually use the same technology to identify whether any 2 individuals, say in this room, share a common ancestor that was most likely in a village in Poland, 20 years ago, because these things go to location. When you start adding in a lot of individuals, that IBD segment has a place. And so that goes more to the entertainment part of genomics, but still, it's important. Anyway, okay, so now I want to get on to real, sort of more prospectively where the field's going. Let me see how I'm doing. Oh, I'm okay. 50 minutes. No, it's okay. All right, so I want to turn now to a couple things that I think exemplify the sort of problems that we have more generally in evolutionary genomics from the standpoint of this new data, not from a purely theoretical standpoint, and talk you through a couple of these examples. They exemplify 2 things that I think are sort of the most promising paths for evolutionary genomics in the near future. One is, of course, to elaborate mechanisms that have to be incorporated into the sort of canonical or accepted ideas about how to think about evolutionary genomics, and what models need to be excluded, because they can be excluded based on all this data. And the second thing is when you don't have any data, as I said before, one of the most pernicious problems that happens is that you don't have to worry about your parameter values very much. And so the scale of things like how big does the mutation rate have to be, or how big does recombination have to be, or how strong does selection have to be, these things have never been taken very seriously because there really never was a lot of data on which to reject or accept any particular domain for those. And that's going to change and is changing very rapidly. And the primary aspect of it is it's going to drive into functional biology the consideration of genetic changes which cannot be detected in the clinic or the laboratory directly. Now, evolutionary genomicists have always been willing to entertain those kind of variation, but biology in general has not been, especially experimental biologists have their paradigm for understanding the biological system, which is to break it and fix it. If you break it and you don't observe any phenomena, then you can't tell whether you fixed it. And so we're in the midst of a sort of transition where experimental biologists and the broader biological community is starting to accept the idea that there may be parameter space that's relevant that is not observable in the laboratory. So let's take it back to the Null Model, the most common model that is accepted and widely used as a beginning for understanding evolution, genomic evolution and population genomics, which of course is the neutral model that most of the variants, or all the variants, or the ones you're looking at, have no functional consequence, and that some random processes, probably random sampling, drive their loss and fixation. And a classic observation, the first thing when it became clear that molecular data when it started to become clear to Linus Pauling actually, was to realize that finally we're starting to have units. One of the characteristics of this period that I was talking about, the 70 years, is you have a science which had essentially no units, no physical units. They had this abstract information unit, which was an allele, but they didn't have any physical units that related to the genetic information that was being transmitted. And with the advent of molecular techniques, immediately the first thing that came out was that, and this is something that Pauling sensed and was formalized by other people, especially Moto, Kimura, is the idea that divergence would be equal to the mutation rate. And that's a very simple idea. You can have the expected number of new mutations. Of course, it's just the 2n is the number of genes in the population if we have diploids, and that's the mutation rate. And the probability that any one of them will be fixed is of course 1 over 2n. They're all equally likely. And so you see that the rate of divergence is equal to the rate of mutation. And if the mutation rate doesn't vary very much, then we have a constant clock-like phenomena. The sister of this equation, which is widely used in biology, of course, is the one for heterozygosity. In other words, random genetic drift removes genetic variation every generation by chance. And if mutations putting it back in, you get this very simple equilibrium equation here that predicts the amount of variation that would be in the population, the average number of differences, say, between two genomes. And one of the earliest things, when the earliest data came in, even before DNA, Dick Lewontin raised this contradiction, and it's part of this whole period of very weak data that this contradiction was basically acknowledged and then ignored for 45 years because we couldn't really address it in any significant way. But when he raised it, the person who, among others, who immediately thought he had a solution to the thing was John Maynard Smith. And Maynard Smith said, well, the answer is very simple. What you have is that in populations, occasionally, rarely, mutations arrive with rare mutations. Of course, since there are mutations, arise, and they're positively selected, and they very quickly go to fixation in 1 over S, where S is the selection differential for the mutant. And as they go to fixation, they drag along a section of the chromosome because the recombination is occurring, but on the scale of the selection coefficient, it sometimes happens and sometimes doesn't. And so around the selected site, you get the fixation of a region of the chromosome that was in this one chromosome, in that one individual that got the mutation. So that leads to a little island, if you will, of reduced diversity. And what Maynard Smith pointed out is in larger populations you have more mutations, and so you would have more fixations. And so more of the genome would look like this, and so consequently the overall amount of variation would never rise very high because bigger populations would keep substituting more of those. Small populations would have few, and so the total amount of variation might be more similar between those two.
So question.
Yeah.
Those stacks.
Oh, excuse me.
Same sequence out
Yeah.
of
Okay. So let me just. Sorry, you very correctly raised a good question. Just imagine these are a region of the genome, and these are all of us. Okay, and some of us have this variant, some of us don't. Okay, and let's suppose this afternoon one of us transmits a chromosome that has a favorable mutant, and then the process goes on, and some 1 over S generations from now, everybody in the world has that mutation, and around it we have a little island of fixation. Okay. Well, this problem has not gone away, even though that was 20 or 30 years ago. And recently, these folks wrote a review paper, where they looked at lots of different species. And the range of heterozygosities that is predicted, and some of these data are not as good as others, and some of the outliers have biological issues that one could raise with them. But in any case, the overall impression is still the same, that it seems quite narrow. And so we have this conundrum that if we think that most of the polymorphism that we see in natural populations is due to mutation and genetic drift, just in and out being lost, then why don't we see a bigger dynamic range of the variability? Because we know the census numbers for, say, Drosophila versus humans is many, many orders of magnitude different. And this is just a contradiction which we have one proposal from John Maynard Smith. There are a few other ones that have been put forward that are also use and leverage good population genetics theory. They're not as well. Some people say they're better supported, but I chose not to present them all this afternoon. Okay, let me put it that way. Okay, so the second thing I want to say, well, how could we ask whether hitchhiking, which is the hitchhiking effect, was as John Maynard Smith called that effect, what's a prediction of that we could test? Well, one of the things that became clear when you did genomic analysis is you could ask two things. If you have a recent hitchhiking event, you can see here you have less polymorphism. So you can see here, since the common ancestor is very recent and there have only been a few mutations, then there are very few polymorphic sites in the region, and the divergence is more or less the same, or a little bit, maybe a little greater. But anyway, it's the characteristic of reduced variation, which is what we were just talking about. But the other thing that you can notice when you have one of these recent sweeps that wipes out all the variation in some genomic region is that the mutants that accumulate after this hitchhiking event, which I represented by this bar, turn out, because of the nature of the genealogy that happens, everybody has a recent common ancestor right there, almost all the branches are leaves, and therefore the mutations are all singletons. And so when you look at the frequency spectrum of polymorphisms, you discover that hitchhiking produces a big skew towards rare variants. And so we can ask about that too. Okay, so we have two things. We expect the polymorphism relative to divergence to be reduced, and we expect the frequency spectrum to be skewed. Okay, and I'm going to, you'll see a figure here. It still has this term here. This is a sort of a test statistic. It's actually the log of the p-value, because the p-values are so large here. I took the log of the p-value, comparing the predicted and observed levels of polymorphism and divergence in windows of the genome. So now we are really jumping to complete genomic sequencing of the whole genome. And this is one chromosome of Drosophila for a large sample of genomes. And the blue line is the windows of polymorphism across the genome. And you can see it drips off here at the telomere. Here's the centromere over here. And it goes back down. Here's divergence, which is more or less flat across the whole region. Okay, so divergence is not changing greatly across the chromosome arm. But polymorphism is changing a lot, both on the small scale and on the large scale. And on the large scale, we can notice right away, and it was noted even before this data was presented, there was already good anecdotal hints, that these regions have reduced crossing over. So if you think about a hitchhiking effect, if recombination is less, then the region affected by the selected locus will be greater, and that's a simple explanation of that observation. And recombination rate also goes down more slowly in this region of the genome, which might explain the reduction in polymorphism there. Now, on a much finer scale, you can see these little regions here where there's been sort of wild reduction in polymorphism relative to divergence. These are very small scale. Presumably, many of them, although maybe not all of them, are local recent hitchhiking events where a favorable mutant went through and wiped out polymorphism. More problematic to interpret are what are these ones that go in the positive direction. This means there's too much polymorphism in those windows. Those have not been analyzed, and we don't have a really good interpretation, although one could make some classic models to use. One could use classic models to investigate those. In any case, there's on two scales there's sort of evidence for the hitchhiking effect. And on a third observation, which I'm not showing you, is that if you look in windows across the genome, you will see that that the frequency spectrum of polymorphism is in fact skewed in this region and down here, and indeed skewed under many of these peaks that drop down. So consistent again with the hitchhiking effect, having the recent hitchhiking event in those windows. Okay, the last thing I want to talk about, which I might make if you could tolerate it for a minute, is this business about weak selection. So most of the genome, if you think about it as a physical entity, is in chromatin. Most of the things that contact the DNA are actually nucleosomes. So here are 4 proteins, histone proteins. They form, there are 8 of them together that form this structure here, and 147 base pairs of DNA wraps around that. This is the primary compaction of the genome from meters down to microns. Okay, and that's achieved by this one interaction. And 80% of the genome is in contact with these 4 highly conserved proteins. The most significant DNA-protein interaction for the genome by far, energetically, space, footprint, everything else, is this one right here. Okay.
Question.
Yeah.
What functions are those proteins serving other than
As far as anyone knows, they package DNA. Now, that said, you see these little tails sticking out here? There are whole departments, recent departments formed around, maybe not around one tail, but maybe 3 of those tails. Okay, so epigenetics takes place on these tails largely, although some of the things in the backbone are also modified. But they play a role in when the genome actually functions. These things have to get out of the way, and they also mark parts of the genome. There's lots of information on those molecules that's not coded in the protein, but is added afterwards. Okay, but the fundamental compaction of the genome involves these very conservative parts of the genome. And these 147 base pair particles are then packaged into higher order structures to achieve ultimately the kind of very strong compaction of the genome that's important. The important thing to remember is whatever happens in the genome, whether you replicate it or you express it or you do anything else, you've got to get those nucleosomes out of the way. Or you at least have to put them on the side someplace and keep them in a special place. You have to do something to get through because you have to unwind the DNA, the double strands of the DNA. You're going to have to displace it. When you isolate these things from the genome and you look at the DNA sequence that's actually in there, there's nothing particularly remarkable about it except what jumps out right away, and it was with the first 183 sequences that were observed more than 25 or 30 years ago, was the dinucleotide periodicity. So, and it's a 10 base pair periodicity, so the DNA, if you remember, turns every 10.4 base pairs. So every 10.4 base pairs, the frequency of a particular dinucleotide, say AA, goes up and down. Okay, and so those AAs interact with the protein in a specific place. And there are other dinucleotides that are also 10 base pair periodicities that interact in other places. So this is just to say what I just said. These dinucleotides are understood from physical chemistry to affect the wrapping of the DNA around. They make the DNA backbone curve around the protein and fit on the protein. They lower extrastatic interactions with particular amino acids that interact. The GC dinucleotide has the opposite effect. It's on the when it's on the inside, it's in the major groove. So if you remember, the DNA helix is not completely symmetrical. There's a minor groove and major groove, and it's on the inside. When it's on the inside, it interacts. And so the question is, what forces shape these patterns? Is it mutation bias, or is it actual function? In other words, are these interactions required to really make a functional good nucleosome? Well, it's been a roaring controversy for a number of years, a decade or more, because one thing is there wasn't actually exquisite data down to the base pair, so people were modeling and they had machine learning models trying to figure out where nucleosomes were, but they didn't actually have the nucleosome sequences in great abundance. And so there was a controversy about which of these processes is important. And so what we did to address this, and this leverages the genome sequencing technology that I'm telling you about, is we isolated nucleosomes, got out the DNA from the nucleosomes, sequenced those nucleosomes, and in the past, people have been too stingy to throw out all the data except the 147. But if you throw out all the data except the 147, of course, you achieve one base pair resolution, and immediately you see, here is the dinucleotide pattern that you see in the DNA, and what you see is the divergence follows the implied pattern. In other words, if you were ancestrally a G in a region where A is preferred, you're much more likely to diverge to an A. And if you were ancestrally a G, an A in a region where G is preferred, you're much more likely to diverge to a G. Now, if you're thinking about this very much, you can say it still could be mutation. The nucleosome should be guiding mutation so that it just fits this periodicity. Why not? It's intimate interaction. Well, this is where population genetics comes in one last time. It's just an example of what one can do with the sort of theoretical constructs that exist. But for example, it's a very simple thought process. If mutation is biased, that's fine, but once an allele arises, it's just any other allele, and its frequency should be the same, whether it arose by a positive bias or a negative bias. Its frequency in the population. And the only thing that could change that frequency is if it had a functional difference that affected fitness. So what one can ask is not only what about divergence, but we can ask what is the frequency, what are the frequencies of alleles that have arisen, the preferred alleles and the unpreferred alleles. Okay. Here, a little bit busier, but for example, if you look over here, here are the GC dinucleotide frequencies in blue, and here is a measure of the deviation in the frequency spectrum for GC polymorphisms, and you can see they line up perfectly. So it's very clear that the preferred allele is more common in those positions in the nucleosome where it tends to be a G or a C in that position, and the unfavored allele is less common. So this is very strongly consistent with the prediction of natural selection. The thing I should say is the molecular biologists, as far as I know, have never found a mutation whose function is mediated through the binding of a nucleosome, especially through the dinucleotide interaction. So it could very well be, and I think when faced with this, many molecular biologists would be willing to accept, do accept, the idea that maybe nucleosomal functions are important, but they're inherently sort of 1% or 0.1%. Actually, biologists can rarely measure 1% in the laboratory. So if we imagine that these things are inherently weak, they're still quite strongly selected. The last thing I would say about them is, if you remember, I said 80% of the genome is wrapped in this stuff. So the actual selective footprint of this is comparable. It's on the order of the same magnitude as coding regions because of the huge footprint in the genome. I'll just leave this up here. This is more or less what I covered today. I don't have time to really go into it, I think. Okay.
So you've given us a marvelous story about how the techniques have to improve.
Yeah.
I see more and more of the details. Can you speculate just a little bit of what might be, the first payouts now for humankind on the whole? What kind of insights might now be possible based on these new techniques, and where do you see the most impact of giving us information on a societal level or medical level?
Oh man, I see. It's hard for me to choose. If you get it like just one drink in me, I start to talk about entertainment genomics is the most important part, to be honest. But I mentioned, maybe it's my intellectual environment that I work in, but the people who are going to be most greatly impacted by genomics, besides doctors and maybe school teachers, I don't know how I feel about that. Certainly law enforcement, I hope less. But anyway, the people who really are going to be impacted scientifically are going to be people who study populations of organisms in nature, because the tools that ecologists and demographers have to figure out what's happened and what's going on in populations, very crude tools. They have to catch and toe clip the lizard and then catch it again. It's really hard work, and you don't get much information. This gives you information on many orders, many time scales, very quickly. You just grind up a bunch of these individuals and sequence them. So I think for people interested in population biology broadly, and it might even extend to human sociology, there's great potential there. I, as a matter of professional approach, I almost never comment on health-related things because it's in good hands, as they say. Anyway, so I guess that's where I see one. I also wonder about, and you guys might have better ideas about this. I wonder whether, as one pushes these things along and tries to get more out of the data, what the sort of tool development or conceptual development will be on the impact. Because historically, trying to understand population genetic variation has had a huge impact on statistics in other areas. And now we're going to enter another era. And so it could be that there'd be quite an effect there, although I have to honestly say I don't see it yet. Michael.
So this idea of weak selection on nucleotides associated with binding to nucleosomes is fine, but a lot of those nucleotides are also doing other things, right?
Yeah.
So those mutations are going to be very constrained.
Yeah.
I mean, encoding regions.
Yeah, everything I showed you was non-coding.
Okay,
But let me just say, you can sort of see it even in coding regions a little bit. But anyway, this is all non-coding or intron. It's either intron or introgenic.
So the suspected function is to latch on the nucleosomes.
What?
The function you suspect of this periodicity is to help latch on the
Actually, if you ask, it's amazing. These are the people who work on nucleosomes. They have first of all, they solved a beautiful structure. It's one of the best structures in biology, and they're really smart, hardworking people. But the actual physical chemistry of nucleosomes is pretty mysterious. The forces are weak, and they say, well, of course they have to be weak because it has to come off and on all the time, but it has to be. The DNA always has to be wrapped in these things, but it has to come off and go on. And so it's thought that it has to do with the kinetics of the actual nucleosome coming on and off, assembling and unassembling. That may be the important functional part.
Kinetics as in velocity or torque?
Yeah, who knows? I mean, there are people, people try to study these things in one can hope that maybe some of these results will interact with sort of physical thinking about what nucleosomes do.
The secondary structure you showed us.
Yeah.
Secondary. How well understood is it? So
Well, there's a very well solved crystal structure for a single nucleosome. Well, for probably 6 nucleosomes, different nucleosome things. They're very high quality, but of course they're not solid. And it's well known that if you look at nucleosomes in solution, they breathe, they come on and off a little bit and everything, but they stay put. I mean, they can slide, they can be moved by remodelers, proteins with energy that can move them around. They do get moved around, but they do stay put and they wrap the DNA. I'm not a molecular biologist, so you can tell. I don't really know what I'm talking about.